All right, everyone, welcome back. We're going to get started here in just a second talking about our new subject, which is metabolism. All right, so one of the things that's really important to recognize about this section is that, you know, first of all, you've had it before, and we're going to cover some new things, but we've talked about fermentation and aerobic respiration in other classes and in cell biology and A&P. So some of it should really be familiar. Some people find it a little heavy going anyway because it is somewhat abstract. It's talking about biochemistry, and you have to love biochemistry to find that easy. And what I want to stress for you is that metabolism is really relevant and very tangible if you think about what it's applied to, especially this growth section that we just went through, how you can use your materials in your world, which ones you can use, what types of conditions are survivable for you, and how successful you are in those conditions depends on your genes, and your genes determine your enzymes, and your enzymes determine your metabolic strategy. So in the interest of time, I'm going to go through some of these questions a little bit more quickly than I normally would. So just a reminder basically of what metabolism actually is a term that sort of encompasses all of our sorry, I had to pull up a different, I just realized I have the wrong PowerPoint here, and I apologize for that. Encompasses all of our biochemical reactions. This is the one that I want. There we go, new share. I'm just going to close that one. Alrighty, here we go. Sorry about that. It'll just take me a second. There we go. All right. All right, so metabolism encompasses all of our biochemical reactions, including the things that break down the nutrients to release their energy and their components, and the things that build up that components using the energy into larger structures. Where energy comes from, different cells get energy from different places, but presumably in the past few classes you've learned that cells like our own primarily use aerobic respiration to get energy. So take organic compounds, break them into pieces, release the energy, use the pieces. And we also have the capacity to do fermentation, right? Especially in our muscles. Really, all of our body can do fermentation. Unfortunately, fermentation releases a lot less energy, so it's not enough to survive on. And that's why you can't survive without oxygen, you as a human being. But when we talk about microbes as a whole group, their metabolism really depends, right? So some of them are like us. They do aerobic respiration and a little bit of fermentation, but others of them don't do any aerobic respiration at all, do only fermentation, and still others use a pathway that you probably haven't studied, which is anaerobic respiration. And so metabolism relates to growth, as I've said, because we need the breakdown reactions of nutrients to produce energy to drive growth. And the growth rate, how fast you can grow, depends on what metabolic strategy you're using and how much energy is created by it. And so we consider all of this important because all of the things we've talked about so far in this field of microbiology, like why we care about microbes, even though they're small and we can barely see them, even using a high-powered microscope, all of those same things apply here because metabolism explains the growth of those things. All right, so what this section covers, and from your background you should know this, is that it covers enzymes and how they work, types of energy, the importance of electrons, and then it goes into detail about the different types of metabolic strategies. Some organisms use aerobic respiration, some use anaerobic respiration, some use fermentation, which is different than anaerobic respiration. And then also In the video lectures, you'll notice that I do a lot of comparing prokaryotes to eukaryotes, which is important because in the past you've probably mostly covered eukaryotes. And the microbes, which do include eukaryotes, also have a lot of prokaryotes. And then the last thing that is not usually covered as well, in some cases at all in your previous courses, is carbon fixation pathways. So that would be including photosynthesis, which you may have covered in cell biology, probably did not cover in AMP because humans are not photosynthetic, and a process called chemolithoautotrophy, which is very unlikely to have been covered before. All right, so we're gonna just go into some definitional stuff. These words in blue are categories that are used to define organisms based on their energy source and their carbon source. Like, where do they get their energy? Where do they get their carbon? Sometimes those two things come from the same thing, from organic chemicals, let's say, and sometimes they come from very different places. So where do heterotrophs get their energy and get their carbon? Write that into the chat for me. Where do heterotrophs get their energy and carbon? Happens to be the same source. Okay, most of you are writing food, which is a good answer. We can be more specific, and some of you are being more specific. You're saying organic molecules, which is a better answer because the food that we eat contains chemicals, organic chemicals, defined as things with a long carbon backbone and lots of hydrogens, and that's how we get our energy and our carbon. Okay, so our energy comes from the organic chemicals in our environment, such as food, and carbon from organic chemicals as well. So we are heterotrophs, so are many microbes, and that category breaks down organic chemicals, releasing the energy, and also then using the carbon as a raw material. Let's look at photoautotrophs. This, of course, would include things like plants, but there are many microbes that are photoautotrophs. So what is the energy source for a photoautotroph? What's their energy source? That's right. Ultimately, the energy source is the sun. Now, the sun is used to actually energize electrons, and then it's the energy in those electrons that's used ultimately in the chemical reactions to build sugar. But the original source is the sun. This energy is used by photoautotrophs to build their own organic chemicals, which requires a carbon source. So what is their carbon source? That's right, carbon dioxide, which is an inorganic chemical. This is not intended to imply that there's two kinds of carbon dioxide, one organic and one inorganic. It wasn't like raised organically or anything. All carbon dioxide is inorganic, so yes. So the carbon dioxide molecules in the air, plus the sunlight from energy, or energy from sunlight, is used to make sugar in these photoautotrophs. Now down here we have this other final category, chemolithoautotrophs. Where did they get their energy? Good. Yes, they get their energy from rocks, which is to say the minerals or the inorganic substances in the world, which are primarily in the form of rocks. And their carbon comes from the same place as the photoautotrophs. They're still going to use carbon dioxide from the air and put it together into these more complex organic compounds that have larger carbon backbones like sugar and amino acids and fatty acids. But the energy that they need to do that process of creating sugar does not come from sunlight. It comes from electrons in these inorganic chemicals. Okay, so if we think about our heterotrophs, and we think about all of the microbes, remember we've got microbes that are bacteria, archaea, we've got some protozoa, we've got some algae. What are the microbes in the heterotroph category? Which of those types of microbes show up here? Write that into the chat. Okay, so many of you are saying protozoa, which is true, but certainly protozoans are heterotrophs. They don't have any way to make their own organic chemicals. They've got to use organic chemicals in their environment. So for sure that's true, protozoa. Some of you are also saying fungi, also true. Fungi also don't have the ability to make their own sugar. They've got to get it from another source. So we've got eukaryotic protozoans, we've got fungi, which are another eukaryote. What's missing from this picture? Bacteria as well, right? Many bacteria. And in fact, I should also for completeness say also many archaeans. So this is a little bit, you know, a little bit lacking. My own list is a little bit short. So fungi for sure, protozoans for sure, all of these guys. Some bacteria, some archaeans. Now, when we think about the photoautotrophs, using the energy of sun to make their own organic chemicals out of carbon dioxide, what are the categories that show up here that are microbial? So you're not allowed to say like, you know, trees, because those are not microbial. Okay, algae, great. Algae are not the only ones though. So we've got algae and some bacteria, correct. Exactly, algae and some bacteria. And some of you also wrote protists. Yes, it would be some protists, right? Because algae are in that group of protists. All right. Now we've got chemolithoautotrophs. And the category here, chemolithoautotrophs, fungi, no. Protozoans, no. It's really only bacteria and, of course, some archaeans, many archaeans actually. Okay, so when I say bacteria on this slide, I mean bacteria and archaean. Okay, good. So here are the different pathways you're responsible for understanding. And the good news is you don't have to know it in the sort of level of detail you'd have to know for a biochemistry class, or maybe even for your AMP in cell biology in terms of like step by step by step. But more you need to understand them at a bigger picture level, like what are overall they accomplishing and how is that impacting the growth and metabolism of the organism. So let's start with just sort of like anchoring ourselves in like some familiarity here. Which of these processes do our human cells use? Write that into the chat. Good. Aerobic cellular respiration is one, fermentation is another. So we do both this one here and this one here. We do not do anaerobic cellular respiration, which for those of you who may not be aware is not the same thing as fermentation. And we don't do photosynthesis and we don't do chemolithoautotrophy. So this is what we do. And we actually have lactic acid fermentation, and then some of our microbes can do ethanol fermentation. So this isn't really what we do, but lactic acid fermentation is the type of fermentation that we're capable of. So now let's ask this other question from your background. You should know the answer to this. If we compare aerobic and anaerobic respiration, what's the difference? So raise your hand if you feel confident to kind of at least get that conversation started. Olivia, go ahead.
Isn't anaerobic cellular respiration it happens in the cytoplasm and the plasma membrane?
Oh, so we're talking about like, is it different in terms of where it happens?
Yeah.
So ish, depending on what your comparison is, but if we stick to microbes only, and even if we sort of narrow down to bacteria only, it's not going to be so much about where it's happening. So that is a good guess. There's better answers for what is the major difference. And let's see who else has raised their hand. Veronica, what would be the major difference between these processes, do you think?
Aerobic respiration uses oxygen while anaerobic does not use oxygen.
Good. So that's true.
Yes.
This uses oxygen, this does not. And then there's so many people that raised their hand. I'm just going to poke one more person. Kelsey, is there anything you want to add to that? So we've had so far that it could vary in terms of where it happens, which is true, but it's a little bit, if we narrow our focus to just like single-celled prokaryotes, not so much the case. We have oxygen versus not oxygen. So Kelsey, what else would you say is the difference between these, if there's anything you'd like to add?
The terminal electron acceptor in aerobic is oxygen, and it's typically nitrogen in anaerobic,
Yes.
or can be nitrogen.
Yeah, so exactly. So that's a great addition to what Veronica was saying, which is that she said aerobic requires oxygen, anaerobic does not. But the terminal electron acceptor is either oxygen here or it's something else here, and often that's nitrogen, although it's not the only option. Good. So here is kind of my little summary of how they're different from each other, and also how they're the same if we sort of compare them. So all of both these processes, because they're respiration, they go through a process of glycolysis, which should be familiar to you, and the Krebs cycle, which should be familiar to you, and the electron transport chain. And then they either end up in oxygen or something else. Now, what the first answer did highlight importantly is that, from Olivia, is that the locations of where both of these processes happen does differ between prokaryotes and eukaryotes. So that's an important thing we'll come back to later, and I appreciate her bringing that up. Okay, so let's look now at anaerobic respiration and fermentation. This is the one that most people struggle with a little bit. Fermentation is something you may have learned about before. You probably haven't learned about this process. However, it will give you two pieces of information to just sort of file away. Both of these, fermentation and anaerobic respiration, are considered anaerobic metabolism because neither one requires oxygen. And I have run into people who incorrectly teach that these are the same because they're not themselves aware. So this is a different process than this, but both of them are called anaerobic metabolism. So with that in mind, how did these two processes that are both anaerobic, so that's something they have in common, how do they differ from each other? Raise your hand if you feel like you want to take a stab at that. Serena, go ahead.
For fermentation, it only does glycolysis. It doesn't do the Krebs cycle or the electron transport chain at all.
Yeah, that's a great answer. So if we look at how things are going here, whoops, I started my animation a little early. I don't know why that's happening. Glycolysis is the process where we get the energy, and then it ends up in pyruvate, which is the end product of glycolysis, you'll remember, and pyruvate is then converted usually to acid or gas. All right. Now, if we compare these two processes, right, they're the same, or they are similar in the sense that both of these are used to do something called carbon fixation. Both of these processes are done by organisms that are able to make their own organic chemicals, usually in the form of glucose. And they do it by taking carbon dioxide out of the air, which is a single inorganic carbon molecule, and putting it into a large organic carbon backbone using different forms of energy. So now my animations are doing wackadoodle things. I don't even know why. If they're the same in that sense, that they're both fixing carbon, they're both making sugar, how do they differ from each other? Raise your hand. Denise, go ahead.
Isn't photosynthesis light dependent, and then chemolithoautotrophy isn't?
That's right. Good. Yeah. So photosynthesis puts carbon dioxide into sugar using energy from the sun, because it's an energy requiring process. Chemolithoautotrophy does the same exact thing, but the energy doesn't come from the sunlight. So it's done by organisms that live in dark places, to sort of simplify. All right. So all of these produce in common. So all of these are considered sort of processes that generate energy. And what form is that energy in? Just write that into the chat real quick to kind of liberate some of those metabolic thought processes from last quarter or however long ago it was for you. Yes, ATP. All of these generate ATP. And here it is. Hopefully this is a familiar molecule, adenosine triphosphate. So here's the adenosine part, here's the 3 phosphate groups. And Lord have mercy, I am so sorry. There must have been an update since I last created these slides, because I've got all sorts of wackadoodle things on the slide. So let me see if I can just go back here for a second. All right. All right, so when we talk about making ATP, the process of making it is called phosphorylation. And there's 3 major types of ATP production. We have substrate level phosphorylation. This is what happens in fermentation. It's also what happens in the glycolysis pathway, and also in the Krebs. If you're not remembering this, both glycolysis and the Krebs cycle produce a little bit of ATP. and they do it by this method. And what this means is just a fancy way of saying they do it in like one chemical step. Like you have a molecule, it's got some energy in it, and you use that energy to make some ATP in like one step. Oxidative phosphorylation is that hugely complicated thing that happens in the electron transport chain. So if you'll remember, like at the end of respiration, the electron goes through the electron transport chain and its energy is used to make ATP. And it's a whole process of generating a concentration gradient of hydrogen ions and then letting them come back through ATP synthase. So that process is called oxidative phosphorylation. It's very efficient, it creates lots of energy. And photophosphorylation is the process of generating ATP during photosynthesis. So we want to think about like where these things are happening. Oxidative phosphorylation primarily happening in the electron transport chain of respiration, and photophosphorylation happening during photosynthesis light reactions. So these slides here kind of just remind us of what those types of ATP production look like. And this is out of your video lecture. In the interest of time, I'm going to kind of skip over this stuff because you can go look at it yourself. But I do want to kind of show you just visually like what I was just saying before, that here are the steps of glycolysis, and you will remember that there's a step where we make some ATP down here, and you can see that there's a chemical, an enzyme turns it into something else, and ATP is made in one step, and same thing happens here. So that's substrate level. If we were to look at the Krebs cycle, we'd see something similar, that there's like a single enzymatic reaction that produces ATP. Here is oxidative phosphorylation. You can see it's much more complicated, right? The electron transporter NADH drops its electrons off, turning back into the oxidized form of NAD+. Those electrons travel through, and every time they travel through, they release energy that's used by these proton pumps to move hydrogen ions into the, in this case, a eukaryotic picture, the intermembrane space of the mitochondrion. Now we have a gradient, and that gradient is allowed to come back passively through ATP synthase, which spins this motor and produces ATP. So complicated way to make ATP, but very effective. Now, if we look at photophosphorylation, the first step of photosynthesis is this light reaction where we have to make ATP. And it works by stealing the electrons off of water, heating them up with some light, and then taking that energized electron and passing it through the electron transport chain, which, just like in respiration, establishes a proton gradient, which is allowed to come back through ATP synthase and make ATP. This picture looks a bit different than the last one, but very similar, except for the, like, where did the electrons come from part. All right. So, as we sort of mentioned earlier, metabolism relies on enzymes. And so I'm just going to ask you, and again, we're sort of trying to like lubricate those parts of your brain that did metabolism earlier and kind of bring that back up. So I'm going to show you a pathway, and I'd like you to write in chat if you recognize what the name of that metabolic pathway is. Good. Yes, this is glycolysis. And what we can see, this is a lovely diagram in my view because it shows us glycolysis in lots of different ways. We can pay attention to it in terms of what is happening to the chemicals. Like here's the starting molecule, glucose with 6 carbons. You add a phosphate, you add another phosphate, you break it in half, you add more phosphates, and then you remove the phosphates, you rearrange them again, and you end up with pyruvate. So you could look at what's happening with the carbons. You could look at the chemical names of all these intermediates and the starting and ending materials, or you can look at these enzymes that are facilitating each of these reactions. I'm not expecting you to memorize all of this stuff, but I do expect you to sort of understand it at this level, right? Not even as detailed as this, but kind of what's happening with carbons, and then also, as we'll talk about later, what's happening with the electrons. All right, what pathway are we in now? Great. Okay, so all of you were alive in the last 2 classes that you had to take prior to coming here, and so you recognize that this is the Krebs cycle, right? So our pyruvate is being dehydrogenated to turn it from a 3-carbon thing into a 2-carbon thing. This acetyl, we add CoA so it can get into the merry-go-round of the Krebs cycle, right? So along comes our oxaloacetate, which is 4 carbons. We add our 2 carbon acetate, we make 6 carbon citrate, and then we rearrange it, remove some electrons on a carbon. We remove more electrons on a carbon. Now we're down to 4 carbons. We rearrange it a couple times, and in the process, we get out more electrons and some energy. Okay, thanks, Becca. All right, now we're in this pathway here. What's shown here? Good. That's exactly right. This is the electron transport chain, right? All the electrons that have been harvested in the process of glycolysis in the Krebs cycle and respiration end up here. They run through this sort of series of steps to remove their energy, use that energy to pump protons across a membrane, and then that gradient that's established drives the production of ATP. So one of the things we want to understand is how enzymes are influenced. We remember that enzymes are made of proteins. Hopefully we remember that, and that they can be denatured, right? That means that they fall apart. Their hydrogen bonds fall apart. We remember that there's environmental factors that can cause this. So write that into the chat real quick. What are the 2 major environmental factors that can cause denaturation? Good, yes. Having too high of a heat or having the wrong pH. Exactly. And then, so those are sort of like environmental factors. It's also possible for there to be chemical factors that influence enzyme function. And sometimes those chemical factors help, but other times they hinder the enzyme, they inhibit it, they prevent it from working. There's 2 main types of enzyme inhibitors that you're responsible for understanding. So write into the chat what those 2 types are. What are those categories of enzyme inhibitors? Good. Yes. Yeah, so we have competitive inhibitors, and then we have what's called non-competitive, also known as allosteric, because it refers to a shape change. Both of those words are acceptable. So this is non-competitive, but also could be referred to as allosteric. When we look at the effects of temperature, we saw this during growth. We saw that growth rates are affected by temperature unequally above and below the optimal temperature. Above the optimal temperature is a very rapid catastrophic decline of growth, and below it's a very slow. And this is related to the fact that enzymes underlie metabolism, and enzymes are proteins. And so as you increase the temperature, you can lead to denaturation, whereas if you lower the temperature, you just cause a gradual slowing in function. And similarly, pH, we remember from growth that the growth curve at different pHs is symmetrical, and that is reflective of the fact that the enzyme function that is allowing these guys to grow is affected equally in both directions as you go away from the optimal pH, whatever it might be. Okay, so if we look at this picture, it's showing us a type of enzyme inhibition. We can see that there's an enzyme, there's its active site, that's where the substrate's going to bind. And in here, there's an inhibitor that's sitting in that active site, preventing the substrate from binding. What type of inhibition is shown here? That's right. This is competitive inhibition. This inhibitor is competing with the substrate for access to the active site. In this picture, we see that that's not the case. The inhibitor doesn't bind the active site at all. It binds an allosteric site, which by its definition means it changes the shape of the enzyme, including its active site, so that the substrate can no longer bind. What type of inhibitor is shown here? Is non-competitive, right? I just asked you and answered my own question. All right, so now let's see if we understand those concepts in these like case studies. So here are 2 chemicals that look very similar to each other. One's called thymidine, one's acetothymidine. The only difference is that instead of this hydroxyl group, there's a little amine group over here. This is a chemical called AZT, and it's effective. You may know this from the medical field, that it is one of the first really important anti-HIV drugs. And what it does is it plugs into the active site of the reverse transcriptase, an enzyme used by HIV to replicate, an enzyme that we as humans don't have. And because of this little NH3, this is the OH group that normally you would add a nucleotide to when you're building a chain of nucleic acids. This is like the T part of the A, G, C's, and T's in the DNA. And instead, because of this amine group, it terminates the chain. So what type of inhibition is being shown here, competitive or non-competitive? What method is this drug using to overcome HIV replication? Good. That's right. It is competitive, right? This competes with this chemical. This is the substrate for the enzyme. This looks really similar. It's similar enough that it can sit into the active site, but then it can't actually do any chemistry. That's the perfect definition of a competitive inhibitor. Why doesn't this drug hurt us? Let's see. Let's have a raise your hand if someone wants to just say, why is AZT by and large not harmful to humans? Emily, go ahead.
Can you hear me?
I can hear you.
We don't have a reverse transcriptase. That's not something we use normally.
That's exactly right. HIV is a virus. This is the viral polymerase that helps HIV to replicate. Because of its unusual life cycle, this is an enzyme that does something that there's no enzyme in the human body to do this chemistry. It takes RNA and makes DNA out of it, not something we do. So this drug doesn't really harm anything that we've got going on. Okay. Remdesivir, some of you may have heard of, is an adenosine nucleoside analog inhibitor. And it inhibits this enzyme called RNA-dependent RNA polymerase. This is another viral enzyme in another type of virus that is used by that virus and not found in the human body. So this is the enzyme used by things like Ebola, but also by things like coronavirus. So this drug was tested for potentially being a treatment for Ebola and currently being tested as a potential treatment for coronavirus, because the idea is that if it inhibits this enzyme, which these guys need, maybe it could be useful. And it works very similarly. Instead of thymidine, it's an adenine analog, but again, it plugs into the active site. So similarly, it's a competitive inhibitor. Now, if we look at something like Zyklon B, which is a poisonous gas, was used famously in gas chambers during the Holocaust and has been used in other sort of terrible incidents in history. We can see that how it works is by inhibiting this enzyme called cytochrome oxidase. So it binds this enzyme complex and changes its shape, preventing electrons from going to oxygen. So then what happens is we get this kind of like pileup of electrons. We can't make any more ATP. And that's really disastrous for people. So is this type of method competitive or non-competitive? That's right. It is non-competitive. Zyklon B doesn't like compete with access to the active site. What it does is it changes the shape of this enzyme so it can't do its job anymore. It's lethal to us because, of course, we require oxygen. And if we can't put electrons on oxygen, then what happens is we can't pass electrons down this chain. We can't set up this hydrogen concentration gradient. And since there's no hydrogen concentration gradient, or the gradient dissipates, this stops working. We stop making ATP. And even though we can still do fermentation, it's not enough for us. Would this agent be lethal to all bacteria? Good. Many of you are saying no, and that's accurate. Okay, and that's because, of course, there's many bacteria that don't rely on aerobic respiration. Jessica, you raised your hand. Did you have a question?
No, I was just going to say it's because some of them don't require oxygen and stuff like that. So
yes, exactly right. So yes, I appreciate you really wanting to sort of take that next step there, right? is to say that's why it's not going to kill all bacteria. For some bacteria, it would be actually a death sentence because they might be obligate aerobes like us. Okay, so there's a number of other things that you can look up. We're not going to do this. If we had more time, we would as a group, but you can look up arsenic poisoning and sort of figure out like, well, does that work the same way? How is that competitive? Is that non-competitive? We can also talk about like the names of some of these enzymes. We can see ubiquinone reductase, cytochrome reductase, cytochrome oxidase. This is referring to electron transfers. And so we're going to start here next time talking about what these terms mean and sort of, again, refreshing our memories of some of these things that you will have learned in the past, but maybe have forgotten or never thought about in the same way that I'll teach it, which is that the reduction oxidation reactions that you may have learned in chemistry are super important in this context in the human body. Okay, so we are out of time. So I'm just going to scooch ahead to the thing I wanted to tell you. I'm going to talk about test 1 on Thursday when I have a little more time. And I just wanted to make sure that everyone is on the same page about what they need to do for lab this week. So you need to continue your unknown ID process, finish lab 3, which involves making some videos, taking a selfie, taking more pictures of your dilution series, making a data table, and analyzing some growth pictures that are provided. And you want to get started on the lab 4 prep. So by Sunday, you're going to need to finish the step 2 of your unknown, make sure you're ready for step 3, submit your lab 3, do your pre-lab 4, and contact your lab mates about lab 4. The last thing I want to say, and I'll sort of recap some of that stuff on Thursday, is that you do have homework for Thursday. Very important homework. Do this homework. There's a metabolism worksheet that's published. I need you to do that and bring it to class because it's just going to kind of get your brain working on some of this stuff you should already know. And if you don't have that, it's going to be pretty confusing to be in the Zoom on Thursday. All right, so I'm going to stick around if you've got questions like I do, and I'm happy to answer those questions. But if you just need to log out, then go ahead and log out. Thanks for joining us today, and I will see you guys on Thursday for more fun, exciting things.